All right. Well, thank you very much, Francis, for this introduction, and also the committee for the honor of inviting me to this meeting. So I'm really very happy to be here, and I'll be speaking quite a bit about pangenome graph and why I think this is a very exciting topic in bioinformatics and for both biology and medicine. But before I start, I want to do a bit what actually Fiona recommended yesterday, Fiona Brinkman, and just to follow on what Francis said in my introduction in terms of just talking a little bit about my background. So as Francis said, I'm from Montreal. I was lucky to start as an undergrad with David Sankoff, who actually introduced me to this community of both ISMB and Recon. At a time it was much smaller than it is now. And one of the first things I learned from David was when I was looking to do a graduate school, he actually recommended that I go to Pavel's lab. And I was quite surprised because the two of them at the time, for those of you who know, were really competing on various area of studying genome rearrangement. And I learned many things from them, but one of the things I learned is how much, this is friendly competition. We're all in this together to really understand some of these big and complex problems. And in the end, again, we're all really working together. After that, I was lucky to have other mentors. I worked in Singapore with Ed Liu, who was the director of the Genome Institute there. Sort of transition away a bit from bioinformatics more into genomics. That's where I started working on transposable element and started my lab. And now I'm back here in Montreal for almost 15 years. Again, I'm quite fortunate to work under the leadership of Mark Lathrop, and I now also have a second small lab in Japan, working with two senior colleagues. So, throughout the talk, I'll talk a lot about some of the work that I'm doing with many of my trainees, but I think I really am quite thankful for all my mentors and people who have guided me along the way. And I'm actually currently on sabbatical in Japan, and so it's also quite a pleasure to be back in Montreal. This is my only trip of the year. All right. So with that context, I want to talk about something that I've been sort of discovering and finding really quite exciting these days, which is really this question of thinking about the reference and perhaps using pangenome graphs. So just a bit of motivation. So I'm sure many of you are working in genomics and are working with data sets that and for which you use a reference genome, right? So one of the first steps is often you take the reads that you obtain from a particular sample, whether it's looking at genetic variants or looking at epigenetics. First step is to map to a reference genome, and that step, is by itself introduces some biases, right? So the reference genome is not the same as the individual samples that you're sequencing. In every individual, there's actually sequences that are missing from that reference. And so this will lead to a bias in the analysis downstream. And so this is an issue in general, but it gets even more acute if you're thinking about samples that are coming from diverse population, different ethnic group, and so on. So again, going back to the example of Japan, so if you're thinking about obtaining samples from a Japanese individual, to map all of these reads onto a reference that doesn't have Japanese ancestry is probably not the best thing you can do. And then on top of that, if you also think about the fact that thinking about the human genome, an individual genome is diploid, again, this concept of using a single reference linear genome is probably not optimal as a first step for all of these downstream analyses that we're doing. So as a bioinformatician, this question of what reference to use, is often an important topic. There's also, there's always different version of the reference that you might be using. But in this case, and that's what I'll be talking about, really changing not just the version of the linear reference, but moving on to a graph reference is what I'll talk about today. So for those of you who were at Evan Eichler's talk yesterday, of course, you'll be quite familiar with many of these concepts already. I will take time to explain a little bit what I mean by the pangenome graph here to make sure that we're all on the same page. And after that, I'll go into more specific examples to show really the benefits of using this new type of reference for both genetic analysis and epigenetic analysis. So here we're really talking about a graph to represent the human genome, a variant graph that actually is going to capture the fact that some segments are shared between all individual, but the structural variants that we've known for many years that are present, we'll incorporate them directly into the reference structure that you're going to be using. So there's nothing new in the fact that there's structural variants in the human genome, but typically we don't use them and embed them in the reference. And so this is what we'll want to do with a variant graph. If you look at a more specific cartoon to make sure that this is clear, so if you have two sequences in a population of genome that you want to represent, you really incorporate that not as a linear reference, but as a graph structure where you can represent what you have on the left, an insertion, a SNP in the middle, and an inversion of a segment on the right side. Once you have the graph structure, then individual genomes or individual haplotype genome represent path on this graph. In this case, you have two paths, the blue path and the orange or salmon path. And in this case, there's actually 8 possible haplotypes, but we're representing 2. So if we try to do that for the human genome, it doesn't completely change what the human genome looks like. You still get that most of the human genome remains linear, but you do capture now within the reference some of these bubbles or some of these structural variants that corresponds to these alternative paths. So you have, this is an example of a relatively simple and easy region of the genome. Of course, towards some of the centromere or the satellite repeat, you'll get quite a bit more variance and complexity. But still true that most of the genome remains linear overall, but now we do have encoded within the graph this genetic diversity. So genome graphs are not new, and I should say that I myself have not been contributing much to the developments of the methods themselves that have been developed over the years to really process and build and align on graphs and so on. This is something that has been developing for decades, especially with in the context of doing genome assembly and so on. But one big change in why the way genome graphs can now be used is the fact that long-read technology, and Evan was pointing this out yesterday as well, long-read technologies are now much more mature, both in terms of the accuracy and the length of these reads. This is really changing the way we can sequence and assemble genome, and all of a sudden, we can really start thinking about building these high-quality genome graphs through the use of these long-read technologies. So I'll go again in terms of just bringing everybody up to speed on this and putting some context for the examples that I'll show, I'll just go a bit deeper into some of the results that were presented in this paper last year, the Draft Human Pangenome Reference paper. So again, thanks to the long reads and thanks to the assembly methods, 2 years ago there was the first completion of a complete human genome, but as technologies go, once the technology is mature, you can quickly ramp up from having one complete human genome to having many complete human genome. And the big contribution of this pangenome reference consortium and reference consortium paper, in my mind, was not so much to really to sequence, in this case, 47 complete human genome, but was really to, again, build a framework that's going to allow us now to use all of these complete human genome in a single reference in a graph for downstream analysis. So again, I'll just highlight some of the key results from that study, to which I participated, but I had only a small role. So as part of the Human Pangenome Consortium, the effort was really to, again, start addressing this question of diversity in the human reference, and identify sample from a diverse group of individual as much as possible, taking into account, of course, a constraint that these references will now become part of the reference and will be publicly released. So, quite a bit of work on just the ethical side of this, and making sure that, of course, these samples were consented for the kind of use that was going to follow. So again, it's a selection of a diverse group of individual, and then the first step was the sequencing. So if you look at the quality of the sequences that were generated as part of the consortium for the reference, so you have on this plot, so on the y-axis is just the contig length, and you see at the very top CHM13, which is the T2T assembly, the first haploid assembly that's really the highest possible quality, nearly complete chromosome across the board. Somewhere in the middle of all of the purple lines, which are all of the complete genomes that are part of the HPRC, you have the reference that many of us are using, the current very high quality human genome assembly. But you can see that in this new set of complete and diverse genome, in purple, these are all genomes of extremely high quality, some of which are even better than the current human reference that many of us use. The next graph is really the one that sort of, impressed me the most from this consortium to some extent was how much new human sequence was being detected in these assembly. Because, so just to go back for a second, so, okay, this is great. We now have 47 genomes, so 94 haploid assembly, but how do we use them? So this is really the step, again, that the HPRC has been working towards, by building a graph that incorporates all of these sequences into a single data structure. But to go back to what I was saying, here now on the x-axis, you see that we're incrementally adding new assemblies to the reference, and each genome is adding significant portion of new DNA that's not present in the reference. Again, to some extent, this is not a surprise, but the scale at which we're getting, if you look, 5 megabase of sequence that is found in 95% of the haplotypes. So this is 5 megabase of human sequence that was basically missing from the reference quite significantly, but is quite common. If you go down to anything that's found in 5% of these haplotypes or more, we're talking about 70 megabase of, again, human sequence. And then each individual continues to add very unique sequences that are missing from the reference. So again, when we just use this simple linear reference, there's all of this space that we're not studying and not looking at. So hopefully making the point that this is going to be quite important. So where are additional sequence being added? A lot of it, of course, is associated with repeats and satellite repeat and low complexity repeat. But you also have regions, and this is typically associated with duplication, that are associated with gene copy number variation. So again, on the x-axis, you have different individual, and then you can see that, in terms of gene content, there's even variability between different human individuals. So if we now look at what that actually looks like in terms of a graph, and now this is the initial example I was showing you were toy examples. Here we're now looking at the real region of the human genome, the HLA loci, where again we know that there's variation, but now we have a way to capture that variation in a reference structure. So all of the loops represent alternative and structural variants in this region. And then on the top right side, what you have are examples of haplotypes in the graph that are also shown at the bottom in the more canonical way, linear reference. So a lot of effort into sequencing these high quality genome, building the reference graph that can be used. What about, again, as part of this initial paper, there was already some demonstration of what you gain now from having that reference. So now, the initial assembly of these genome was done using long reads, but now you can go back and look at the analysis of short read data using this reference versus the linear reference, and look at the impact that it has on improvement on even short read and small variant calling. So there's already a gain, and without again going into too much detail, there's already a gain, and I'll get back to where this gain is coming from a bit later on. So there's, even by using this new reference, there's a gain in short variant calling. There's an even bigger gain, of course, and I think this is where, in terms of, like the significant differences, especially in structural variants. Structural variant calling using, a combination of the linear reference with short read has really been playing with so many false positives that they were very difficult to use. I mean, over the years, there's been tons and tons of work in terms of building better and better algorithms to call structural variants from both short read and long read to some extent, but it was still, I think, quite error prone, making the analysis and the usage of those calls quite challenging. But now, as part of this, what they were showing is that you really basically double the amount of structural variants that you're able to call. If you see, it's mostly, I don't think you see my mouse, but overall you double. And this is especially, of course, not so much in the easy region of the genome. I showed you some examples of regions that are mostly linear, and so you don't get a huge gain in easy regions of the genome. But in regions such as tandem repeats and so on, is where you're getting the biggest boost in terms of being able to call structural variants in a way that I think now means that they can really be analyzed and used in ways that they haven't in the past. So that's really sort of the introduction of what I wanted to present. the fact that we didn't have all of these regions present in the reference, the fact that we couldn't really study structural variants at the same level, were we missing things in a number of downstream analysis for both genetics and epigenetics. So I'll now move to this part of my talk. So the first project was really a collaboration with a good collaborator, Tomi Pastinen, who used to work with me at the Genome Center here at McGill, but has since moved to Kansas City. And Tomi, I think, really had the vision a number of years ago when long-read sequencing was still quite expensive to gamble that for a rare disease patient, and in many cases where unsolved cases of rare genetic disease, that perhaps the long-read technology would actually bring new information to try to understand the variants that are contributing to disease. So from a cohort of 10,000 subjects, they identify a number of difficult trios and probands for which they applied long-read PacBio sequencing. And so I came in later after that with my student Christian Groza, who just graduated, to try to see whether using the graph approaches on this data set could really help us identify structural variants that of importance in this data set. So again, at this point, the kind of graph that you see or cartoon that you see on the left should be familiar to you. This graph also shows the kind of information that's very difficult to encode using a linear graph and VCF, which are more complex regions that have embedded variants within structural variants. So in this particular project, we sort of expanded on the HPRC work and created our own pangenome graph using assemblies coming from these probands. So what you see in red is basically the reverse of the graph I showed you from the pangenome paper, which is the number of sequencing. As we add genomes to the graph, the number of sequences that we're adding that are not present in the reference. So we start with the reference. After that, we start adding all of the HPRC genome in red. So we're adding sequences that are not present in the genome, and then we continue when we add, all of the sequences that are missing, that are coming from the specific proband that we sequence. And you see that, of course, the rate at which we're adding new sequences decreases, but we still, anytime we sequence a new individual, we're adding sequences. And in this particular context, especially because we're interested in rare variants that might be contributing to rare disease, these, we believe that, within these individual-specific or quite rare sequences, there might be some interesting disease-causing variant. We did some validation, of course, but using, for instance, we did a number of things. We had twins in this data set, and so we can just look, for instance, at the amount of shared structural variants that we can detect using the graph. So this using the mini graph tool versus, of course, with long reads. Given that these were sequenced with long read, we can also use long read SV caller like PBSV, but using the graph, in this case, we're able to detect even more shared variant. And on the right side, you see a similar plot showing that in terms of SV that we're able to detect and are shared between siblings, we were able to increase the rate. So we really think that all of this has to do with the fact that we have a higher sensitivity using the graph. Once we've built the graph, we can start counting allele frequency and look for, what I mentioned are rare variants. It's quite small. I mean, you can focus on the left side of this graph. So we can, again, we have a number of individuals. We can look at variants that are shared and are found between our cohort, our disease cohort, GFRK and HPRC. Those are not the variants that we're interested in here. We have variants that are only found in the HPRC individual. And then the most interesting set for us in this case were the variants that were specific to our disease cohort at the bottom. But again, these are now structural variants that we are able to call with much higher accuracy and that we can follow up on. So we take the set that's found at the bottom here and look at their annotation. Of course, you know, they're distributed across all over the genome. We can annotate them with gene. We can annotate them with genes that are known to be associated with disease, the omen genes. One of the things that I would argue, though, is that at this point, we're still sort of filtering based on what we know, based on genes that we know have already been shown to be implicated in disease. You know, I suspect that, again, now that we have better, to use a word that Evan used yesterday, a better microscope, I suspect we'll also be able to discover new genes using these kinds of approaches. In this first study, that's not what we focused on, though. We focused on, you know, the ability to use this approach to identify structural variants like the one that I'm showing here that is, you know, is found and is associated with a gene that's already been previously reported to be associated with disease and is a very likely and reasonable candidate here. You know, one of the things is that it's not like it helped us solve, you know, 100% of the cases that we couldn't solve before. And actually, this particular variant was also buried in the set of variant that we might have been able to detect using previous approaches. But the big difference, again, is the accuracy and the amount of structural variants that we now found that, you know, we can trust and we know we can follow up on. So I really think that, again, this will be quite helpful in terms of identifying new genes and variants in more difficult regions of the genome that we can follow up on. For the conclusion for this part is that using graph, we can obtain a higher level of reproducibility of structural variants compared to standard reference approaches. You know, we have large sets of structural variants, but as I said, I think much easier to follow up on some of these candidates given the quality of the calls and the data. So this is the example I wanted to give on using pangenome graphs to help in genetic analysis. I actually, in my lab, do a lot more work on epigenetics, and so I'll spend the rest of the time talking about some examples looking at epigenetics and what we can gain from using a graph. So when Christian started in my lab, and this was already a number of years ago, this was actually prior to all of these efforts from the pangenome graph. And at that time, we were just playing with the idea of using personalized genome to see whether having, again, an individual genome, would that actually improve ChIP-seq and other epigenetic analysis. And so if you look on the right, this is from one of our first papers on this topic. Again, you know, using a graph representation, but at the top, what you have is a representation of the linear reference as a graph. And it's quite small, but what you can see on top of that are ChIP-seq reads, actually, and the fact that, you know, we don't get a lot of signal in that particular region. At the bottom, it's quite small, but we now have a graph representation that includes some SNPs and indels. And all of a sudden, because we're using that graph representation, we're actually able to map many reads into that region, and that's the cluster that you see in green, and call peaks in that region that we would have missed otherwise, because again, we were losing reads that should have been mapping to this region. So this was our early work on this topic. And more recently, again, working with Tomi Pastinen, we wanted to expand on this and test more systematically whether, having personalized information from individual, having their actual genome sequence would change our analysis of epigenetic data set. So this particular study was a very large collaboration, again, with Lewis and Tomi, where we took sample from individuals from different ancestry. And this gets back at the idea that, relying on the single ancestry reference genome is probably not the best thing we can do. So in this particular study, we really selected individuals that were either European descent or African descent, and profiled their cell before and after flu infection. And the interest of Lewis was really to look for genetic variants that are associated with epigenetic change and epigenetic response. My interest in the project was, again, going back to this idea that, what's the difference between using a reference versus using these individuals' genome themselves to analyze this very rich epigenetic data set that includes RNA-seq, ATAC-seq, and ChIP-seq that's summarized at the bottom right. So what Christian was doing, this again started a number of years ago. The data that we had was not long read from these individuals, but was 10x, link read data set. So specifically what we were interested in this particular context was to see whether we could look for mobile element insertion, so transposable element, using the link read data set. So Christian devised a method to identify this time not just SNPs and indels, but also mobile element insertion from this data set and the link read data, and incorporate them in the graph to see how it would change our epigenetic analysis. So the first step was to both genotype and assemble as many of the transposable element polymorphism that we could detect in this cohort. So we could detect many T polymorphism. We could assemble, again, at that time, a large fraction of the ALU. The L1 were more challenging. So in this particular data set, we couldn't really fully resolve all of the polymorphism that we know are probably in that data set. But we felt this was already a pretty good starting point where we had a large list of assembled mobile element insertion and polymorphism coming, and again, it's tiny, but coming from all of the known active families, recently active families, mainly ALU, young ALU and young L1 families. So we knew at the time we didn't have a comprehensive set of structural variant in this cohort, but we already had a pretty reasonable set of variants. So we took, this is a summary of all the number of variants we were able to call, both in terms of SNPs and indels in this cohort, and the mobile element insertions on the right. You see that based on ancestry, of course, there's huge difference, as we expect, in terms of number of variants that we detect in the two subgroup that we profile in this case, with the African descent individual having many more variants. So what we did is we took all of the variants that we detected in our individual and we incorporated them. Again, we didn't have long reads, so we didn't have assembly for these genomes, but we had all of these variants and we incorporated them into a graph to see, how it would change our epigenetic analysis. So I'm just showing, one of these examples of what we get with calls from one of these histone marks. So I'll walk you through this one since I think it's an important one. So if you start at the bottom in blue and you have, again, on the x-axis, all of the different individual, you have all of the call. There's two methods. Either we use our graph or we use the reference. And in blue is all of the peaks that we detect using both methods and that are the same. So you see that the vast majority of peaks that we call in all these individual, are the same. But what you see at the top is that you see in orange and green peaks that we can only detect using the graph-based approach, or peaks that we can only detect using the reference-only approach in green. So, I remember quite clearly how disappointed both Christian and I were initially, especially in the previous paper, when we found, it's only 2 to 3% new peaks that we're able to detect using this approach that we've been working on for so hard. Until we took a step back and thought a little bit about that, because okay, it's only 2 to 3% of these of new peaks, but the new peaks that we detect are all the peaks that are enriched for SNPs and mobile element insertions that we have in this case. So both in this paper and the previous paper, what we showed is that the peaks, of course, that are changing are peaks that are associated with genetic variants, or at least are enriched for that. So one could argue that it's a small number of peaks that are changing, but it's the ones that are genetically variable, which really, I think, makes them quite interesting, even though, and to some extent, the fact that the majority of things don't change is to some extent expected. So again, we can zoom in on some of the cases that we're now able to detect. So this is an example from this, that we can detect a peak in a region that's associated with an ALU polymorphism. So again, just to help you navigate through this plot. So on the left side, you have a loop. So the individuals, 29 of the individuals, so we've profiled 35 individuals. 29 of these individuals have an ALU polymorphism, have an ALU insertion there. That while the ones on the right don't have that loop, don't have that alternative path which corresponds to an ALU. And then there's the two conditions. I mentioned that there's before the infection, which is at the bottom, and after the infection, which is at the top. And what we see, and this is relative to, I'm not sure I remember which mark, I think we're looking at ATAC-seq here, is that we only detect ATAC-seq signal, of course, in the individual that have that particular ALU polymorphism, and only in the ALU in the flu condition. The neat thing with this is that I think typically, we do these analyses sort of sequentially. We look for variants on one hand and polymorphism on one hand, and we look for epigenetics, the data on the other, and then sort of we try to combine those two after the fact. But I think, the strength here is that really we're because the polymorphism is embedded in the reference, we're really analyzing the effect of these polymorphisms directly in the analysis of epigenetic data. This does also highlight, and I'll get back to that at the conclusion, is that, it is a bit of a challenge to figure out how to visualize and how to use this new structure. We're so used to projecting everything on the linear genome, it becomes a little bit difficult to look at this data. Again, this is an example just trying to visualize some of the data, but I think a lot of work needs to be done there. So in that particular study, we did a lot of work because we tried to detect the variants and then analyze the data. The reason we ended up joining the HPRC consortium was actually because as we were working on this paper, we realized that instead of using the graph that we had been constructed, we could now use the graph that the HPRC had been working on. So in the results I showed you, we were using our own graphs with the variants that we had detected. But if we use the HPRC genome, we actually recover very similar, the equivalent of a 2 to 3%. This is what we see on the left side. Because most of the variants, of course, especially the common variants that we were detecting in our graph, they're already embedded in the HPRC reference pangenome graph. So actually, I mean, for better or worse, we no longer need to do all the work that we did to build our own graph. We can just rely on this new reference graph, and we get the same 2 to 3% gain in new ChIP-seq that we couldn't detect before. We also just showed on the right side that looking at so this is looking now at allele-specific binding sites with sites that are heterozygous, and then number of reads that we get on these sites that are actually on one of the two alleles to really sort of improve the detection of sites that are allele specific. So again, I think from my perspective, this is just the beginning of what is going to, where, there's going to be a lot of work and then a lot of exciting discoveries potentially from using this richer reference for epigenetic analysis. The final example that I'll talk about is a follow-up to the first project that I described to you that was on the genetics detection of rare genetic variants. Another benefit of the long read sequencing is that together with great information to be able to assemble genome is that you can get epigenetic, and in this case, methylation information from the PacBio HiFi. So a subset of the probands that had been sequenced with PacBio sequencing had methylation data as well. So we actually ended up having 435 methylome, in part because the methylation processing option wasn't turned on when that particular project started. But basically, so we were interested to see whether, a bit like what I just showed you with the ChIP-seq analysis, we could start analyzing methylation data on the structural variants that are specific to these individuals. So very similar to what I started with the pangenome adding a lot of sequences that are missing from the reference. If we now think about the methylome and CPGs, well, there's a lot of CPGs that are missing from the reference, and now all of a sudden we can really profile CPGs that are absent from the reference and look at their methylation state. So these are slightly complex plot, and I know the font size is small, but maybe we start with the main messages here. So we're adding, there's roughly 30 million CPGs in the human genome. We're adding 15 million additional ones. So again, this is mind-blowing. Many of these, of course, are associated with very rare variants. Maybe looking at the methylation in these, rare sequences is not so critical, but it's just, just so much additional data that we haven't been looking at. Because we didn't have methylation on all of these individuals, in this particular case, we were actually able to only genotype in our sample 8 million of those 14 million, but it's still quite significant. So we suddenly have access to 8 million CPGs that we haven't been looking at before. We can observe that, over 5 million of these end up being methylated in at least one of the samples that we've profiled. The right side shows a similar kind of cumulative plot showing that, of course, anytime we sequence a new individual, we discover new sequences and we discover new CPGs with or without methylation that we can study. I'll just show you a few examples from this that we can do downstream. Again, I think there's a lot of exciting and both bioinformatically and on the biology side questions that we can look at. But there's a lot of work that needs to happen in terms of thinking about how we can navigate this new data that's no longer on the linear reference, but on this graph structure. But just to give you a feel for this, on the left of this heat map, what you have so this is a Kerr locus, so this is a bit like HLA, a region that's quite variable between individual with loops that are, and structural variants that are present or absent in these different individual. So light gray, or I can't even tell myself, but light gray or dark gray is reference or alternative. So some of these section of the Kerr locus are found in all individual, others are variable. And then what you see in the actual heat map is the methylation state of this. So we can really analyze, in a systematic way, the methylation status of regions that are either found in all individual or some of the individual. And again, I think there's quite a potential and discoveries that can be made from that joint analysis now of structural variant together with chromatin or methylation states. We can look a little bit at, again, what's contributing these additional CPGs that are missing from the reference. Repeat, my favorite topic. We can look a bit more again at how much of these missing CPGs, and you can see that many of them are actually associated with no repeats. So a lot of this is probably associated with large duplication that again are contributing CPGs that are missing from the reference. And then of course you have a lot of CPGs, additional new CPGs that are embedded in some satellite and simple repeat. But you're also getting, analysis of CPGs on ALUs and L1 that are missing from the reference, and you're getting this all at once. On the right side is a first pass analysis looking at, how much different if you look at canonical CPGs that are in the reference versus some of these new CPGs that we're calling SV CPGs because they're associated with variants, and whether the proportion of how many are actually methylated based on the different types of annotation. So again, I mean, this is really just an overview of some of the kinds of new things that we can look at systematically in terms of annotation of the missing parts of the missing reference. Even more meaningful, I think, and again, this is work that I think is just starting. We can look at sort of meaningful association. This is now looking at trying to see whether we can get new quantitative trait loci that are associated with some of the structural variant. So by combining, again, regions of the genome that we're missing from the reference and trying to see if this was missing some association of genetic variants that were actually associated with a chromatin or a methylation phenotype. So again, I mean, this was to give you, a bit of an overview of the kinds of things I think that we can do by combining both the graph analysis with the epigenetic analysis. So in conclusion, so I think that this pangenome reference really represents an amazing opportunity for, on one hand, to explore new biological questions, especially in it's not going to change a lot of things in the very easy regions of the genome, but if you had an interest in tandem repeats, transposable element, also ability to look into centromeres and telomeres. So I think there's a lot of potential new biology to be found in those regions now that we have a better microscope, to use Evan's expression again. But for this particular crowd, I think, I think this is also extremely exciting time because, thinking about the development of new algorithms to really make full use of this graph structure, I think is really wide open at this stage. I think there's just so many questions of what we can do better in this graph space versus the linear space. I think, I didn't really touch on this a whole lot, but I think the question of annotation in the graph genome, I think is a big challenge. I think aspects of visualization and again, how to navigate these data in this new space is also quite a challenge. One thing that I touched on a little bit in the beginning, but I think is also quite important is I work mostly with, human geneticists these days, and the idea that we want to be more equitable in our analysis and have better representation of different population when we do genetic analysis. I think this really gives us a nice tool now where we no longer have an excuse, and we really, I think, need to embrace, even though there's additional complexity, this type of data structure, because it is less bias in all of the downstream analysis. Just a small pitch for one of these new tools. So Christian and Clément, research associate who was in my lab, we're developing a tool specifically for the analysis of transposable element using graph structures, and really building on some of the work in the papers that I presented today. So if you're interested in this, I would encourage you to take a look at that. Another point I want to make, though, is that, well, this especially applies in some other audience. I think in this audience, I think hopefully all of you are willing to embrace the graph. But I think one thing that's also very important is that actually you can use the graph even without fully moving to the nonlinear genome and to the graph structure. You can very easily just change the reference at the mapping stage and get a lot of the benefit of that more complex, high-level data structure. And once you're in that space, you project, liftover style back to the linear reference and just continue with your analysis. So in the very beginning, what I showed you with the gains in both small variance and structural variant calling really just comes from this mapping step that benefits from using the richer reference. And if you think about it, is because if you're mapping in the richer space, you actually have a lot of additional information on true mapping quality and true mapping ambiguity. And so that translates in everything you do downstream, even if you're using the linear reference. So again, if you're not willing to completely go to the graph for all of your analysis, I would at least recommend that you use it at this mapping step. So I'll almost end here. I do want to thank especially Christian. Christian is the student in my lab who, started me on this graph genome. He's actually the one who's taught me everything I know about graph genome. So if I get a tough question, I might have to request a phone call to him, because again, he's been amazing. He just graduated and is now starting a postdoc here with Guillaume Bourque in Montreal, and Clément and Shun, who also helped quite a bit in this work. I also want to congratulate Francis. You have to look up. I want to congratulate Francis and Michel for, so we had yesterday this celebration of bioinformatics.ca. So 25 years, I think, of amazing impact, I think, in terms of bioinformatics training here in Canada. And I really look forward to the next 25 years. I'm not sure I'm going to help for that whole time, but, maybe a little bit. So thanks a lot, Francis and Michel. So this is, I think, very exciting going forward. I wanted to also highlight if you know of students who might be interested. So we have this exciting PhD program between Kyoto and McGill University, especially for quantitative methods in genetics, but also epigenetics. So if you're interested, this might be something to think about. Another plug, I'm doing all sorts of plugs. This is another one. I didn't talk about some of the work that I'm doing in Japan, but this is a very exciting institution there working on human biology, organoids, and I'm actually recruiting an assistant professor, senior postdoc there. So if you're interested in epigenetics and quantitative method, please look for me. And then since Francis talked about the Pan-Canadian Genome Library, I'll just finish with a couple slides on this, just to say that, as was mentioned, this is a bit sort of a very different topic, but about, the idea that, we have many projects in Canada generating human genome sequences And we're now tasked to come up with a good strategy to archive long-term these data sets and make them available quite broadly to the community. This is a project that involves, I'll just skip to that, but that involves a large group of people across Canada. And so I'm quite excited to try to put together something that's going to really maximize the use of human genomic data in Canada. So with that, I'll stop and take any questions if you have.
Yes, go ahead. You have a question?
Guillaume, fascinating even for someone who's a structural protein person, very inspiring. TRIM25 piqued my interest, and I'm aware that it's involved in innate immunity, but I would like you to expand on what are the possible functional consequences of those variations near it.
Yes. Well, thank you. So in a way or in a bad way, the example that we highlighted actually to some extent was already known to be an important region. There's not so, like I ended up identifying a few examples in this particular condition of a new peak that we could detect over a polymorphism. I would lie to say that there's like thousands of these. There's only actually a few small number of variants that, because it's a combination of having both a variant and then observing functional data in that particular condition. So we end up with a small number of regions like this. But I think, again, even though it's a small number, these are variable between individuals, perhaps have an impact. So there's an LU insertion near the promoter of that gene that probably modulates a little bit. Not that the gene otherwise is not regulated following infection, but that probably tunes a little bit, the level of response in some of these individuals. And so, we didn't pursue that much further. I think it's also clear that they're not huge numbers, but I think in the past we would look at this region. We knew, for instance, already that particular region was a QTL for response to infection. What we didn't know is exactly how mechanistically this worked or this didn't work. So I think having now this kind of information where we can combine structural variant information and the chromatin really just allows us to understand the region mechanistically much better in terms of what's happening. But in order to do that systematically and easily, I think we'll need some of these new tools that I mentioned that really allow us to visualize and annotate those regions properly and all of that. So I'm not sure I answered your question completely, but thanks for your question.
Thank you. Maybe in the left microphone here. Yeah.
Thanks a lot. So I had just a question on how do you envision the future of graph outside of human? So first, to study within species variations of other species.
Yes.
And second, for modeling between species variations, do you think graph would make sense?
Very good question. So I focused on the human, but the graphs are already being used quite a bit across the different species and in other areas as well. So there's lots of development. I mean, I focused on human just because that was my interest, but there's already a lot of work in other species. And indeed, if you look at, for instance, in mouse, there's a lot more structural variants between different mouse strains. I mean, everything I said here definitely applies for other organisms. But your question of whether this can also be used between species is very good. I think this is where, again, there's a lot of work that needs to be done because, the parameters of, how much, what distances, is where are you going to get help from graphs and to what level of distance. If it's too different, then I think it's probably going to break down. There's still lots of questions about technical questions in terms of the optimal way of building some of these graphs and how to do that. So again, I think it's wide open question, but I think there's definitely a lot of potential, even within, for instance, again, looking at the kinds of things we're doing within primates to really help some of the comparative analysis that we've been doing the graph structure might be a real better way to do these analyses, because otherwise you're trying to combine lots of pairwise analysis, and that's just not the right framework, I think. And this perhaps provides the right framework.
Thanks.
Thank you.
Sally's going to do an online question.
So we have an online question. Annalika Nwashili says, thank you, Dr. Bourque, for this insightful talk. I'm curious on how the use of a pangenome reference graph enhances transcriptomic analyses, particularly in identifying and interpreting alternative splicing events and gene expression variations across individuals of different ancestries. Has there been any application of the pangenome in this regard?
Excellent question. I think so. I didn't touch on that at all because we haven't done any of these analysis. There's a bit of analysis looking at alternative transcripts. But, you know, alternative splicing to begin with is very challenging and difficult. And now the long reads are also, you know, providing a very powerful tool to start looking at alternative splicing. But again, if you think about combining the analysis of structural variants with alternative splicing, this becomes a very challenging technical question for which the graph really provides the right substrate to provide good answers. So I haven't followed as closely the work that's happening there. I know there's a bit of that work that's happening, of course, within the HPRC, and I would encourage you to take a look at what they've done there in terms of methods. But absolutely, I think that's a great area, and it's tied to what I mentioned in terms of the annotation. So again, all of the gene annotation, which is quite challenging on the linear reference, it to some extent makes much more sense in a graph structure, but it's also quite challenging to organize.
Okay, we have time for one short question.
Thank you so much. I was wondering if you could comment a little bit on how the move over to these sorts of graph-based references affects the challenges and opportunities with respect to separating away from some of the other fields that do sequence-based analysis. So there's a lot of overlap between sequence-based analysis and comp bio and, say, natural language processing. And it seems like moving to graph references means a completely different set of tooling and challenges as these areas sort of split off.
Well, that's a very good question.
Long question.
It's a long question too, for which I'm not sure, but I agree with you that I think it is an area of potential convergence indeed, where other methods could then be coming in, such that the methods working on graph could now be applied perhaps more easily to sequence analysis and some of the things. But yeah, I would have a hard time answering that question in more detail, but I think it is another interesting direction to pursue, absolutely.
Okay, well, thank you very much.